thank you very much for that kind introduction. And it's a real pleasure to be here at UCLA. So what I want to do today is give a pretty broad introduction into the topic for those of you who might not have followed it closely over the years. And then talk about some work in the lab that has been focused on adult stem cells in mice. And if there's time at the end, tell you a little bit about a new project we're undertaking to try to study aging in humans. So this work began about 25 years ago in the lab, and it was driven by two new graduate students at the time, Brian Kennedy and Nick Austriaco. Brian is now the president and CEO of the Buck Institute in Novato, California. And Nick has gone on to do other things in life. But both were very creative and driven students. And they really made this early work lead to something interesting. And what we were studying in those days was aging in budding yeast, which is what my lab worked on at the time. And we chose this system because we thought it was tractable and we thought it was interesting. We certainly didn't think we were going to find anything generalizable about it. But you know, whatever you choose to work on, it should be something that is doable. So that's why we chose it. And the way this works in budding yeast is that the daughter cell is new, the mother cell is old, and what you do is follow this mother cell in the microscope and push away the daughter every cell division and simply count how many times the mother cell can divide. And this was a system that was well established, and it was known that mother cells could divide roughly 20 generations or so. And then they senesced, and they senesced with a characteristic phenotype. They were enlarged, sterile, also wrinkled. So that was the model of aging that we were working with. And we spent about 5 years trying to identify genes that controlled this process, such that if we mutated the gene, it would change the lifespan as defined by this metric in a significant way. And the most interesting finding that came out of these studies was the identification of a gene called Sirtu as an anti-aging gene in yeast. So this is a survival assay of mother cells. And you can see for the wild-type strain, the number of generations is on the x-axis here, and there's this precipitous decline in viability at about 20 generations or so. In this case, it's actually about 30 generations for this strain. And the importance of Sirtu is illustrated by the fact that when the gene is ablated, that lifespan is shortened. And when the gene is overexpressed, here, 2 copies instead of 1, the lifespan is longer than the wild-type control strain. So that sort of focused our attention on this gene. And we continued working on it in yeast. And a paper only 4 years ago from another lab, Jaswinski lab, did a study that was surveying all the genes in the genome for importance in dictating lifespan, and the top of their list was Sirtu. So we've sort of stumbled into an important gene, at least as far as yeast aging is concerned. Now, in the years since, a lot of studies have been carried out in other systems, including the roundworm, flies, and mice. And in these cases as well, it turns out, again, I would not have predicted this or expected it, but it turns out that Sirtu homologs, and they've been called Sirtuins, can extend lifespan when their activity is increased in these systems. So that means Sirtuins have a broad effect to slow down aging and to extend lifespan. So we were interested very early on then in what Sirtu did, okay? And its history comes from something called silencing in yeast. And actually, Mike Grunstein here was one of the players who identified the importance of histones in silencing. And that importance is that the amino terminal tails of histones H3 and H4 have lysines that tend to be acetylated on the epsilon amino group for active chromatin and are deacetylated when the chromatin is silenced. And Sirtu is somehow was known to be involved in this silencing process. So that suggested right away that Sirtu could be a protein or a histone deacetylase, but many attempts to show that in multiple laboratories failed. So we started working on that, the biochemistry of Sirtu, and we purified the yeast Sirtu protein and the mammalian ortholog of Sirtu, and we're studying it for several years, and came upon this activity, which is an NAD-dependent deacetylation of targets. And so what this means is that without the NAD cofactor, Sirtu and Sirtuins in general are inactive, and NAD is actually a co-substrate and is split every reaction cycle concomitant with deacetylation of the substrates for Sirtuins. Now, those substrates can be histones, and in yeast, the histones are certainly critical substrates of Sirtu. But in mammalian cells, in addition to histones, substrates include other proteins in multiple cellular compartments that can be deacetylated and that are important physiologically. So Shin Imai, my postdoc, and I were excited about this finding because it provided a link between protein acetylation, metabolism, and epigenetics. It also had implications for aging because it gave a possible explanation for how a low-calorie diet or calorie restriction might extend lifespan and slow aging. And we proposed that it worked by activating Sirtuins. And I think that hypothesis has proven to be robust, as I'll get to in a few minutes. So with respect to acetylation then, this was the third and final, I think, class of enzymes shown to influence the protein acetylation level in cells. The first was the histone acetyltransferases, identified by David Alice. Second was the HDAC proteins, which are deacetylases, identified by Schreiber and colleagues, and these have no cofactor requirements. And the third are the NAD-dependent deacetylases, or the Sirtuins. So in mammals, and my talk today will focus on mammals, there are 7 homologs of the yeast Sirtu, which is shown here on this phylogenetic tree. The closest is called Sirti1, shown here. And I'll talk mostly about Sirti1 today. And in many senses, this is the most important of the 7 mammalian Sirtuins. And Sirti1, along with Sirti6 and 7, are nuclear proteins. Sirti1 tends to be in the nucleus deacetylating many targets off of the chromatin, and Sirti6 and 7 tend to be more closely associated with the chromatin, suggesting that histones might be particularly important substrates for them. Now, what's interesting is that another 3 of these proteins, Sirti4, 5, and 3, are encoded in the nucleus but target to mitochondria. So 3 of the 7 Sirtuins reside in the mitochondria, and Sirti2 is cytosolic. So is there a rhyme or reason to why there are 7 of these proteins? Are they redundant? No, they're not redundant. For one thing, they reside in different cellular compartments. One way to view this is to look at Sirtuins with this color key here and proteins that they're known to deacetylate. And many tissues have substrates for Sirtuin proteins, shown here. I'm not going to go through this all, but let me just point out PGC1 alpha as an example, because this is a protein that's deacetylated by Sirti1, and which, when deacetylated, can drive mitochondrial biogenesis and mitochondrial function. And in fact, if you go through this list, one of the themes that emerges is that what Sirtuins do in a coordinated fashion is they drive oxidative metabolism in mitochondria, okay? And so they force the cell to produce ATP via electron transport in mitochondria. Now, concomitant with that, they drive stress resistance to reactive oxygen species. So enzymes like superoxide dismutase are upregulated by particularly Sirtuin in mitochondria. They also drive pathways that would be consistent with oxidative metabolism, such as oxidation of fatty acids inside of mitochondria. So I think that's kind of an overarching principle. And note that oxidative metabolism would be particularly important under conditions when the fuel source is limiting because of the efficiency with which ATP is produced by mitochondrial metabolism. So what about this idea that sirtuins are necessary for the effects of calorie restriction? Well, there's a pretty vast literature that is in the hundreds of papers. I will choose one example here because it's particularly clear-cut. And the subject here is hearing loss in mice and calorie restriction. So what these authors did in this study is to examine hearing loss in 12-month-old mice on the control diet, shown here. So what you can see is that it takes a much higher decibel level of sound to elicit a response in the old mice over a broad range of frequencies, shown here. And they're not even that old. These mice will live past 2 years. So this is kind of midlife. And already their hearing is severely damaged. And it's damaged due to oxidative destruction of hair cells in the cochlea of the inner ear of these animals. Now, what the study shows is that if the mice are age-matched to the 12-month-old, but on calorie restriction, they're completely protected against hearing loss. So that's good. That's kind of in line with what calorie restriction does. It slows down the ravages of aging in many ways. And the point of the slide is to show what happens in a Sirtuin knockout mouse. Now, note that this mouse has almost no phenotype to sort of casual analysis. It's just like the wild type. But if you look at it in this assay, you can see that the knockout mice is completely refractory to calorie restriction. Calorie restriction doesn't work in this mouse, and hearing loss is observed. So that's one point about the sirtuins, oxidative metabolism, especially under conditions of limiting fuel sources. And one reason that might slow aging, again, is because of beefing up resistance to oxidative stress and oxidative damage in cells. Now, an unexpected feature emerged more recently, and that is that NAD and sirtuins are intimately connected to the circadian clock. And this was a field that started about 6-7 years ago. And the way this works is the following. So this is the oscillator that is present in the brain, but also in peripheral tissues that oscillates with a diurnal cycle. And the nature of the oscillation is that this is a transcription factor called BMAL and clock, a dimer. And this transcription factor drives the expression of these feedback regulators here, which build up and inhibit the transcription factor. And this occurs with a cycle, and when the transcription factor is inhibited, then the cycle is reset, and this oscillates with a period of about 24 hours. Now, this wouldn't do anything to metabolism at all if it couldn't reach out and affect proteins that are not a part of the oscillator. And what these papers show is that one of the critical output genes that's not a part of the oscillator, but that's regulated by BMAL and clock, is a gene encoding this enzyme, NAMPT. And this is the rate-limiting enzyme for NAD synthesis. And so what that means is BMAL and clock oscillate, and therefore expression of NAMPT will oscillate, and therefore NAD synthesis itself is circadian, and it peaks at the right time. And that means sirtuins are circadian in their activity, and I won't go through this, but there are also more connections between SIRT1 and SIRT6 and the components of the clock itself. So there's this really close connection. And I think that part of the reason for this is that metabolism needs to be staged in a temporal way. And if it's not, things go awry. And there are many instances of, in mice, if you damage genes encoding components of the oscillator, the mice have bad health. In humans, there's considerable data that people who live lifestyles, for example, shift workers, that mess up their circadian clocks are not as healthy, much less healthy. So that's another dimension to this, is that there's a considerable input into this system that makes metabolism circadian. Now, what about diseases? And again, I'm still in this introduction part. In rodent models, multiple sirtuins have been shown to be protective against a series of diseases: cancer, diabetes, neurodegenerative, cardiovascular, osteoporosis, kidney disease, inflammation. And the list is longer, in fact. So let me just say a few words about cancer, where 3 of the sirtuins, T3, T4, which are mitochondrial, and T6, which is nuclear, have been found to be lost in roughly 20 to 40% of human tumors. So that means they're tumor suppressors. And how do they suppress, how do these proteins suppress cancer? And one of the major mechanisms, I think, relates to cancer metabolism, and particularly to the propensity of cancer cells to want to favor glycolysis, which is the Warburg effect, but also glutamine utilization. And so the connection then to the sirtuins is shown on the left part of this slide. So SIRT6 represses glycolysis and also ribosomal biogenesis. And those are 2 pathways that cancer cells need in order to grow. In the mitochondria, SIRT4 suppresses this critical enzyme for glutamate utilization, glutamate dehydrogenase, and thus represses glutamine metabolism. And SIRT3, shown here, represses several enzymes, again, this is by deacetylation, that are involved in ROS production. So SIRT3 suppresses production of reactive oxygen species. So when any one of these 3 proteins is lost, then the cell's metabolism is more geared towards tumor metabolism, favoring cancer. Now, on top of that, SIRT1 and SIRT6 are intimately involved in DNA repair. And that includes both single-strand break repair and double-strand break repair. And I'm not going to go through this in detail, but it includes repair at telomeres. It includes non-homologous end joining and homologous recombination. So again, the removal of one of these 2 sirtuins would create higher DNA damage, which again could favor a tumorigenic phenotype. Now, the last thing I want to tell you in this introduction is something that was found still more recently, and that is NAD is depleted with normal aging. So this is a study that was done in mice. So basically the data is very clear-cut in roundworms, in mice, and emerging data exists in humans. And what's shown here is young versus old mice and NAD levels, very simply, in young versus old liver, and over here is muscle. And there's about a 2-fold reduction in NAD levels. So you say, that's not very much. Is that enough to do anything? And this is a blot of PGC1 alpha acetylation in muscle of young versus old mice. And what this shows is in the old mice, PGC1 alpha is hyperacetylated. And the deacetylase that works on PGC1 alpha is SIRT1. So this implies that this reduction in NAD of 2-fold is enough to deactivate SIRT1 in these old mice. Now, I'm going to move on. We could talk about why NAD declines, and if someone wants to ask a question later, I'll be happy to tell you what's known about that. But it does decline. And the real exciting part is that this is actionable. And the reason it's actionable is you can replenish the lost NAD by feeding the animal NAD precursors. And again, there's a whole series of papers that show this. And again, I want to credit Shin Imai, who first noticed this. And what he noticed is that in a Sirtuin transgenic mouse, the mouse had a phenotype, okay, which was improved glucose tolerance. But when the mouse got old, it lost that phenotype, okay? But the protein was still overexpressed. So he wondered why that could be. And what he hit upon is NAD levels were plummeting in the old mice. And if he supplied NAD in the form of this NAD precursor called nicotinamide mononucleotide, he could restore NAD levels and restore the phenotype to that mouse. Now, it turns out that just wild-type mice, when supplied with nicotinamide mononucleotide or nicotinamide riboside, NR, have an improvement in their health. For example, they're more resistant to becoming diabetic as old mice. And there have been other health benefits described. So this then leads to a notion that therapeutically, one can think in terms of activating sirtuins for health in two ways. One would be NAD replenishment, as I just mentioned. And the other way is something that was found still earlier by David Sinclair and colleagues, that there are certain small molecules that can activate SIRT1, such as resveratrol, and more recently, compounds that are being developed at GSK that will be in the clinic in about a year by binding to an allosteric domain in the protein. And these compounds, the stacks, will target SIRT1, but NAD replenishment will replenish the activity of all the sirtuins. Okay, so that concludes the introduction. Went a little bit long, so I'm going to have to speed up. So I'm going to talk about adult stem cells. And the stem cells are these intestinal stem cells. So this is how the gut is organized. It's these finger-like structures called villi, and the base of the villi, which are called crypts. And the stem cells are at specific positions within these crypts. And they can divide to give more of themselves or to differentiate, to give all of the differentiated cell types of the gut, which move up the villi and are sloughed off very rapidly. It's a very rapidly dividing tissue. Now, the point of departure in this analysis for us was a paper published by Yilmaz and Sabatini a few years ago. And they were studying the interaction between the intestinal stem cell and a niche cell called the Paneth cell. And what they found is that calorie restriction repressed the activity of TOR, and in particular, a complex of TOR called mTORC1. And that led to the Paneth cell synthesizing this small molecule, cyclic ADPribose, which was then sensed by the stem cell to promote self-renewal in the stem cell. So that means calorie restriction gave rise to an increase in stem cells. So the study carried out by Masaki Igarashi in my lab then utilizes mice that are either wild type for SIRT1 in the gut, shown here, or overexpress SIRT1, shown here, or are knocked out in the gut for SIRT1, shown there. And what we did is we looked at the effect of calorie restriction on all of these mice and tried to piece together what was happening, particularly in the stem cells. So here's an introductory slide that shows a cross-section of the gut in wild-type mice, ad libitum versus calorie restriction. And as Yilmaz saw, we saw an increase in the size of the crypt due to an increase in the number of stem cells, shown here. When this was done in a villain-crine knockout mouse that's specifically knocked out in the gut, you don't see this increase. And the data is shown here. For wild type, the crypt size increases, and the knockout doesn't increase. Now, the key experiment is this one here, where we're staining for this marker of stem cells to actually get a count on the number of stem cells. And what you can see is that there's an increase in the number of stem cells in calorie restriction, which is shown here, and that does not occur in the knockout mouse. We wanted to have a backup way to assess this. So we used a mouse now where the stem cells are marked with a GFP, driven by a stem cell-specific promoter, LGR5, shown here. And again, calorie restriction gives rise to an increase in the number of stem cells. And if you knock out SIRT1 in the stem cells, this time with tamoxifen, you don't get that increase. So that backs up the histology. Next, we looked at the overexpressing mice that have higher levels of SIRT1 in the gut. And these mice now are fed ad libitum. They're not calorie restricted. And what you can see is the changes that normally are induced by calorie restriction are induced simply by overexpressing SIRT1. So this mimics the calorie restriction phenotype. You get the increase in the crypt, shown here, and you get the increase in the number of stem cells per crypt, shown there. So this is very nice. It says both loss and gain of function mutations in SIRT1 show that this protein is necessary and its activation is sufficient for the response of ISCs to calorie restriction in vivo. So the in vivo is, I think, a pretty good foundation. So we wanted to know, what are the relevant cells for this? Is it the stem cells or is it the Paneth cells? And one way to approach this is to purify the stem cells and the Paneth cells, which can be done by marking them appropriately and then sorting them in the cell sorter. So you have now a pure population of stem cells and a pure population of Paneth cells. And then you can cultivate them in this matrix. And they give rise to these colonies called organoid colonies. I'll show you a picture in a second. This is developed in Cleaver's lab. And what you can do then is add a defined number of cells of one type or both to a microtiter well and count the number of these organoids that are formed. So these are the organoids shown here, and they are seeded by a stem cell. So a stem cell initiates the formation of one of these colonies, and then they differentiate to give all the cell types of the gut. And they're thought to be an ex vivo assay for the function of ISCs. Okay, so let's look at an experiment. If you co-culture stem cells and Paneth cells, and the Paneth cells come from an ad libitum-fed mouse, you get this number of organoids. But if the Paneth cells are from a calorie-restricted mouse, you see an increase. This is what was observed in the Yilmaz paper. Okay, this was the bedrock assay, that calorie restriction is activating the Paneth cells. Now you can ask the experiment, well, what if the stem cells, but not the Paneth cells, the stem cells come from a knockout mouse? No response. Okay, so that says you need SIRT1 in the stem cells in order to get the response to the calorie-restricted Paneth cells. However, if the Paneth cells come from a knockout mouse, they work just fine. So SIRT1 is required in the ISCs, but not the Paneth cells, to get the effect of calorie restriction. So we began then to focus on the stem cell and to piece together the pathway of signaling. And again, I'll go through this briefly since it was published a month or two ago. Now, cyclic ADPribose, the signal from the Paneth cell, is known to stimulate calcium release in the target cell. So we looked for many pathways that were calcium dependent. And we found that CAM kinase was important in this signaling pathway. So here's an example of that. This is mixing stem cells and Paneth cells. This is the effect of calorie restriction here. And if you use a specific inhibitor of CAM kinase, you block that effect. And a nice control to this experiment is, instead of using calorie restriction to drive the increase in organoid colonies, you could use the SIRT2 overexpressing mouse, right? Remember, that mimics calorie restriction. And when you use that mouse, then the inhibitor doesn't do anything. And so what that means is that the way we interpret this is CAM kinase then is downstream of calcium signaling, and SIRT1 is downstream of that. And if you have a gain-of-function mutation in SIRT1, it's now independent of upstream signaling, and you get the increase in organoids. The other nice thing about CAM kinase is one of its known targets is AMP kinase, which is known to respond to calorie restriction in many settings. So we took a look at AMP kinase in the crypts, and you can see that phosphorylation, which equates with activation of the kinase, is induced by calorie restriction, shown here, and it's induced in the SIRT1 overexpressing mouse, shown here. And furthermore, if you use an inhibitor of AMP kinase, then this increase by the Paneth cells in calorie restriction is blocked. And if you use ACAR, which is an activator of AMP kinase, it's sufficient to give the increase. So this, I think, firmly places AMP kinase in this pathway. And lastly, AMP kinase is known to regulate expression of our friend here, NAMPT, the rate-limiting enzyme for NAD synthesis. And in fact, calorie restriction shows a twofold induction of NAMPT. So we can now trace a pathway that looks like this from, in the stem cell from CAM kinase through AMP kinase, NAMPT, NAD, and Sirtuin activation. That's what we believe. And the question then is, what's downstream of this? So remember, the output of calorie restriction is to expand the number of stem cells. So we looked at known pro-growth pathways, such as PI3 kinase AKT or beta-catenin. And both of these in other contexts, for example, colon cancer, play an important role in driving growth. But they're not in play in the case of calorie restriction. We also looked at mTORC1, okay? And in particular, the signaling from mTORC1 to phosphorylate S6 kinase and to phosphorylate S6 to drive protein synthesis and cell growth. And we found that, bingo, this was the guy. And it's a very surprising result, because I told you earlier, and I'll come back to this, that mTORC1 is canonically repressed by calorie restriction. And here it's upregulated. So is this true? Let me show you some of the evidence for that. Okay, this is looking at the relevant phosphorylation event in S6 kinase, ad libitum, or calorie-restricted mice, looking at CRIPS. This is the same experiment in S6. Again, this is with CRIPS. We did the same thing with purified stem cells now. Okay, this is now an immunostaining experiment looking at phosphorylation of S6, wild-type calorie restriction. You see the increase is very evident, shown here. And from a knockout mouse, you don't see this increase. And you can also see it, and this is a little bit heroic, in a Western blot on isolated stem cells. Calorie restriction gives an increase in S6 kinase phosphorylation at the relevant phosphate, but not at a non-TOR regulated serine. S6, same thing. Now, what's really interesting is another substrate of mTORC1, which is called 4-EBP, is not affected by calorie restriction. So somehow the activity of mTORC1 is parsed, so the calorie restriction is giving an increase in phosphorylation of one substrate, S6K, but not another substrate, 4-EBP. How is that possible? And we really were befuddled by this for some time. And then a paper came along, shown here, that showed that there was crosstalk between Sirtuin and S6K and TOR. And what they showed in the paper is that Sirtuin actually deacetylates S6K. And that makes S6K a better substrate for mTORC1. So we wanted to see if that mechanism was in play. And it is. If we look at the effect of calorie restriction, shown here in CRIPS, we can see it leads to the deacetylation of S6K. And that requires Sirtuin, not shown here. Also, if we look at the overexpressing mouse for Sirtuin, it leads to the deacetylation of S6K. So what we believe is that calorie restriction induces Sirtuin deacetylation of S6K, this residue, and that makes it a better substrate for mTORC1, and that's why the signaling through that part of the pathway increases. So here's what it looks like then, shown here. Sirtuin is upregulated by these upstream steps I mentioned, deacetylates S6K, and this leads to protein synthesis and expansion of the ISCs. Now we also believe, and I won't show data for this, but we could talk about it, is that the actual nutrient sensing here is done by the Paneth cell, which is what senses that calories are low. And the stem cell then is entrained by the Paneth cell via cyclic ADPribose. And mTORC1, which normally would be suppressed in calorie restriction, is not suppressed. It stays the same, but its output is increased because of Sirtuin activation and deacetylation of S6K to make it a better substrate for TOR. Now, what about functional data? Is this true, or is this just fantasy. So if this were true, then blocking these downstream steps in the TOR pathway should have real consequences. Okay, so here's our experiment with organoids. Again, the control, the calorie-restricted Paneth cell, give the increase, organoids, and that's blocked by an inhibitor of S6K, and it's blocked by rapamycin, the famous inhibitor of mTORC1. What about in vivo? So we've done multiple runs of this experiment in which we calorie restrict the mice and either treat them mock or with rapamycin and ask what is the effect of rapamycin. Now note that prior to this study, one would have assumed that rapamycin itself would be a mimic of calorie restriction because that's how it was thought to act, and does act in many cases. But let's see what happens in this case. Okay, so this is the data here. Here's the tabulation. Okay, this is the crib size. Calorie restriction gives an increase, blocked by rapamycin, shown there. And here's the bottom line. You count the stem cells, and calorie restriction increase, and it's blocked by dosing the mice with rapamycin. So this is interesting because Sirtuin and mTORC1 are usually working in opposite directions. So the genetics says that Sirtuin upregulation slows aging, and for TOR, downregulation slows aging. During aging, Sirtuin activity is thought to decrease, which is bad, and TORC activity to increase, which is bad. And calorie restriction does the opposite. Sirtuin activity increases, which is good, and TORC1 decreases, which is good, but I just showed you that's not true in the ISCs. And a therapeutic then that would mimic calorie restriction would be something that would activate Sirtuin, the NAD precursor or the stacks, or would inhibit TOR, rapamycin. But one would predict that that would not be a good thing for these stem cells and for the gut. Okay, now what about aging? So everything I told you so far relates to diet. Okay, these studies were all done with young mice. What's the effect of normal aging on the stem cells in the gut? So we did that study, and here's the data. So I'm just showing you the data on counting the stem cells shown here in young mice versus old mice, and the number of stem cells goes down from about 5 per crypt to about 3 per crypt. So that's consistent with what we think about adult stem cells, that they decrease in number with aging. Now, interestingly, if we look at by immunostaining or by Western at signaling through the pathway I just described you, so the pathway I described to you relates to calorie restriction. It may or may not play a role in normal aging. We don't know. We wanted to query whether it did. And in fact, if you look at S6 staining with aging, it goes down in the old versus the young gut. And by Western, you can see this clearly. Phosphorylation of S6K is down. Phosphorylation of S6 is down. And importantly, the levels of NAMPT go down with aging. So that's the stem cells. What about the Paneth cells? Did the same thing, young versus old mice. And surprisingly, we did not see any change in the number of Paneth cells in the old mice. It stayed the same. No decrease. We could also look at the function of the stem cells and the Paneth cells by the organoid assay ex vivo. And I just look here. If we mix together young stem cells, or stem cells from young mice with Paneth cells from young mice, you get this. If you mix old, you get that. There's a clear deficit. Okay? But here's the revealing part. If you mix old stem cells from old with Paneth cells from young, deficit. But if the stem cells are young and the Paneth cells are old, no problem. So by this functional test as well, the problem with aging is in the stem cells. So the stem cells seem to be taking the hit and not the Paneth cells. And is this reversible? So we looked by the organoid assay. Again, young versus old. There's a deficit in the organoid formation. This is just the stem cells now. And if we supplement with the NAD precursor, NR, that's rescuable. Okay, so that's consistent with the idea that NAMPT is down and that everything downstream of that in the pathway is going to be down. So NR can rescue that. Furthermore, that rescue by NR is blocked by rapamycin, the inhibitor of mTORC1, and it's blocked by EX527, which is an inhibitor of Sirtuin. Do the same experiment with now, this is with CRIPS, with purified stem cells, same result. NR rescues the old purified stem cells, and that's blocked by rapamycin. What about in vivo? The study here was then to take young versus old mice. And let's just look at this, which is the stem cell number, which is sort of the bottom line. And old, you see a decrease in the number of stem cells. I showed you that earlier. And that's rescuable by NR in vivo, which is really surprising to me that you could actually rescue that. But I think nice. So the model then for aging now is that the same pathway is in play here, and that aging leads to a deficit that we can trace at least as far up as NAMPT here, which means that NR can come in at this level and bypass the deficit. And the deficit leads to a decrease in mTORC1 signaling and a decrease in ISC number and function with aging. Okay. So again, if we return to the canonical view, the genetics, what happens during aging and calorie restriction and therapeutic, we can see that in every case, what happens in the ISCs is the opposite of what one would predict by the canonical view of mTORC1. So it's possible that the ISCs are the only exception to the canonical view, or it's possible there are more exceptions that are yet to be uncovered. So to summarize then, this part, the aging part of the talk, is that the wiring of pathways important in aging can be cell type specific. And I just showed you that the stem cells seem to be wired differently. And the implication is a drug to treat aging globally may affect different tissues differently and even oppositely. And mammalian aging is complex. Okay. So there's a little time, so I'm just going to take a few minutes to shift gears and talk about a new project. What I'm interested in these days, not yeast so much anymore, or worms, or even mice, but I'm interested in aging in humans. And part of that interest is of everything that's been found in the model systems, how much of that would carry over to humans, if any. And the other part is there a way to directly interrogate humans to learn about aging? So this is a study to study aging in the human brain that is in its early stages, being led by a postdoc in the lab, Kristen Glorioso. And it's a collaboration. And it's a collaboration with computer programmers, okay, which include Andreas Fenning and Manolis Kellis, MIT, and other collaborators that supply us with tissue and data. And these are centers that have collected banks of human brains, okay, which is what you need to do a study of this sort. And I'll note that these first two banks are disease-free, so quote unquote, brains. And this cohort of David Bennett is weighed towards Alzheimer's disease, some Parkinson's disease, but also some control, disease-free. So here's an example of the cohorts and the age distribution. The Pittsburgh cohort has a nice distribution, many different ages here. NIH, pretty nice. ROSMAP, the disease cohort, is very narrow age distribution, skewed towards old. And GTEx, I'm not going to say anything about today. So the region we're looking at then in this study is the prefrontal cortex, and it's a particular region of PFC called BA47. And the reason for that is that the brain is so heterogeneous with so many compartments that you have to focus in on one specific region. Otherwise, it's almost impossible. So what we do then is it's very simple. We look at genes that change as a function of age. And of course, there are genes that go down, like this, BDNF being a pretty important protein for brain health, and that go up, such as GFAP, shown there, and identify these aging-regulated genes, of which there's roughly 1,000 or so in the Pittsburgh cohort, and most of those overlap with similar changes in the other cohort. Okay, so that's good. And we use this data then to develop a metric of what we call the molecular age of the brain by, and I don't have time to go through that, but basically we try to break the data down into principal components and throw out the principal components that seem not relevant to aging and weigh the principal components that seem relevant to aging and develop this metric. So what does it look like then if we, let's say, compare the molecular age of the brain, as defined in this way, with the chronological age of the subject at the time of death? Okay, so here's that data for the Pittsburgh cohort, molecular versus chronological age, molecular here, chronological there. And as you'd expect, there's a pretty good correlation. Okay? But what we wanted is what you see, a correlation that's good but not perfect. And the reason is we want to take for each data point here is a brain, the difference from the regression line shown here and the actual data point to define as a variable called delta age, okay, which is the molecular age minus the chronological age. And that tells you how different that brain is from the average. So something above the line here is a brain that's older than the average, and below the line is younger than the average. And some of them are older or younger by a fair amount. And it's a continuous variable that applies to every single brain. And we take that as a proxy for the aging rate in these brains. Fully aware we're not measuring rate because this is not longitudinal. This is all cross-sectional. Nonetheless, we take this measure of delta age as a proxy for the aging rate. And our goal, long-term, is to identify genes and pathways that determine this aging rate and determine why this brain is aging fast and this brain is aging slow. Okay? But what I'll just give you a little tiny vignette today is how this data, this proxy for aging rate, what it would tell us if we compared a cohort that had Alzheimer's or Parkinson's brains versus disease-free control brains. What will we see? And let me call your attention here. So this is from the ROSMAP cohort, and these are the control brains here. So on the y-axis is delta age. Minus means molecularly younger than average, and positive is older than average. And this is the Alzheimer's set there. And what you can see is there's a significant difference between the mean, and the Alzheimer's brains are significantly older molecularly than the controls. Okay? You can extend that to looking at amyloid levels in the brain, shown here, or tangles. So again, younger brains now on this side, older brains on that side versus amyloid levels, and significant correlation. The younger brains are more protected against amyloid. Same for Parkinson's. Okay, so this would lead to a hypothesis then, one hypothesis, and there are others, but one hypothesis would be that people whose brains have age more slowly are more protected against Alzheimer's disease. And conversely, people with a faster rate of brain aging are more susceptible to Alzheimer's disease. Now, many of you would say, well, that's obvious. And we knew that already. But I think this is a rigorous demonstration of that. Now, it becomes interesting also when you compare this to the known major risk factor for Alzheimer's disease, which is the ApoE4 allele, okay, which, as far as I know, stands way above any other risk factor for non-familial Alzheimer's disease. There's nothing else even close. And I'll show you a little data here then that shows the relationship between delta age and ApoE4. Okay? So we took the cohort and divided it into groups that significantly deviated from the average delta age. So this is -5 years is the youngest, I think, decile in the cohort in terms of aging rate, and +5 years is the oldest decile, highest aging rate, and the number of ApoE4 alleles is shown here. What you can see is quite remarkable that this big risk factor for Alzheimer's, what's plotted here is the relative risk of AD in the cohort. Having one allele of ApoE4, normally a very significant risk factor, is blunted almost to insignificance in the young brains. So what we interpret this to mean is that slow brain aging can suppress having an ApoE4 allele, and that these two variables of brain aging and ApoE4 are functioning independently. Conversely, in the older brains, you can see that it's much higher with no ApoE4 alleles. There's a higher risk of Alzheimer's, and the effect of ApoE4 is very evident. So one way of thinking about this, then, is ApoE4 is a risk to Alzheimer's, and rapid aging is a risk for Alzheimer's. And we also, I didn't show you the data, but we asked whether the ApoE4 brains, compared to control brains, showed more rapid aging, and they didn't. They were the same. So what we think, then, is that these two risk factors are independent, roughly similar magnitude, and together is the worst combination leading to Alzheimer's. I list other phenotypes here. I think our data is strongest for Alzheimer's disease. So, and again, what we want to use this tool for is to probe more deeply into the mechanism of brain aging and what determines it. So with that, I'll stop and take questions, and I just will acknowledge my funding sources, disclaimers here, and Masaki Igarashi, who did all the gut work, and Kristen Glorioso, the work on the brain. Thank you.